redox potential, you already know redox potential ability to hold electron. Oxygen has very high ability to hold electron, or it has very high affinity to electron. So this is called redox potential. Okay, this is called redox potential, and number is given for that redox potential. If some element has very high redox potential, it is given more positive number. If some element has very high redox potential, very low redox potential, it is given negative number. So you can see this NaDH is given negative number, and this has oxygen is given positive number. So this oxygen is very capable enough to suck electron from NaDH. You can see here in electron transport chain. If you can see in electron transport chain, I will show the electron transport chain here. So electron transport chain, here you will see oxygen here, and here you will see NaDH. So this chain from NaDH electron flows like this, okay, and here, and it will reach oxygen. What does oxygen do? Oxygen actually suck the electron from this, because oxygen has very high capacity to hold electron. So this flow happens because of sucking of electron. You can understand that way. When you turn your concept to your own daily activity, you suck your Coca-Cola, you suck. So in that way, it sucks the electron, and that is how electricity is created here. And because of this electricity, proton, you can see proton pumping here. Proton is being pumped here, and this proton will produce ATP. This proton will be responsible for rotation of turbine. You must have seen, when in order to rotate turbine, we make a dam. So this inner mitochondrial membrane is a dam, natural dam. Okay, proton cannot cross through this inner mitochondrial membrane. It will have to cross through a turbine to rotate the turbine. So you can see this number are given. You don't have to worry about this number. Just remember, you can see positive number higher. This number will increases. That means as you go from complex 1, from complex NaDH, as you go from NaDH to oxygen, from complex 1, complex 3, complex 4, you will have high redox potential, means high affinity. Affinity will increases. Affinity increases as you go from complex 1 to complex 4 to oxygen. So you can see in complex 1, and for 1 you can see this FADH2, and these are, we will talk about complex. So complex 1 will suck the electron from this, and it will transfer to ubiquinone, and ubiquinone to this, because of this increasing redox potential. So you can see here, the electron transport chain. You can remember this way. From NaDH, the electron goes into complex 1, 3, and 4. 134, 134, and here from FADH to 234, 234, 2 to 3 to 4, 2, 3, 4. So you can remember 2, 1, 1, 3, 4. In case the electron movement will be from 1 to 3 to 4 in case of NaDH, and in case of FADH, this electron movement will be from complex 2, complex 3, and complex 4. Just memorize this: 234 and 134, 2, 3, 4. So I will tell you where this flow of electron from NaDH will reach oxygen. Here also it will reach oxygen. Okay, this will make a circuit for flow of electron. Because of this, proton will be pumped. Now here I told you, do you remember? I told you complex 2 is nothing but succinate dehydrogenase. Succinate dehydrogenase is called complex 2. And you know, in succinate dehydrogenase, what happens? Succinate is converted into fumarate, and this enzyme will produce FADH2. And then this enzyme directly transfer electron to coenzyme Q here. So you can see here the flow in between two complexes. There will be coming something else here in between complex 2 and 3, and complex 1 and 3 also. Before 3, you can call before 3. Here also before 3, you can see coenzyme Q. Coenzyme Q is coming. So I will talk about what is coenzyme Q. I will show you what is coenzyme. So this coenzyme Q will come, and this coenzyme Q will take this electron that is present in FADH2 complex 3 here in case of FADH2, and here from complex 1 to complex 3. Okay, so this is how coenzyme, and then between complex 3 and complex 4 there is a cytochrome C that is present between this is inner mitochondrial membrane, and this is outer, between inner and outer mitochondrial membrane. So when the electron flows from this complex 1 to coenzyme Q to complex 3 to complex 4 to oxygen, complex 3 to complex 4 through cytochrome C, complex 1 to complex 3 through coenzyme Q. When electron flows like this, because of electricity, you know this is electricity, and electricity, because of electricity, you must have seen when electricity flows, it switch on the bulb. So here, what, instead of switching on the bulb, proton is pumped. So because it has energy is liberated from the electron, that energy is utilized to drive this proton pumping, and this complex 1 has proton pumping mechanism. So complex 1 will pump 4 proton, remember, per 2 electron, because from NADH 2 electron is transferred. So from per 2 electron 4 proton is pumped. Now again, if you see in case of complex 3, here also you will see 4 proton, but when you see in case of complex 4, you will see 2 proton. So 4, 1, 4, complex 3, and 2, complex 4. But when you see here, complex 2 does not pump proton. Zero pumping by complex 2. So that is what you have to remember. 1 pump 442. You can, in case of NADH, you need to remember 442. In case of FADH, complex 2 does not pump. This is 0, 4, and 2. So only 6 proton is pumped because of FADH2. 2 plus 4. Only 6 proton. Here you can see 10 proton pumping. So now you can find the meaning why NADH is equal to 2.5 ATP. And this is called 1.5 ATP, because and energetically this electron has less energy. That is why it will pump less proton. This NADH electron has more energy. That is why it will pump more proton. So this is actually the difference. It has got less energy, less proton, more energy, more proton. So thermodynamically, if you see energetics, this is the region. So memorize this. FADH pump 6 electron, and this NADH pump 10 electron. Because why 6 electron? Because complex 2 does not pump proton. Only complex 3, 4, complex 4, 2. Okay. Now we have studied till now we have studied the function, what this complex do. So we will talk about, because I have told you these complexes are like wire, like this wire. At the center there is metal. So we will talk about what is this wire and what is this metal, rather than we will talk more about this metal part. Okay, so which is conductive part of this molecule. So you can see conductive part of this molecule. If you can see here, this protein, there are protein, this wiring is protein, so cover is protein, and this conductive part here in complex 1 is FMN and iron sulfur center. So iron sulfur, I told you, in place of copper you will see iron here. So FMN and iron will be the wire of this complex 1. And from this complex 1, in between this complex 1, this is also a part of wire, this coenzyme Q between complex 1 and complex 2. Now in complex 3, the wire is formed by cytochrome B and C. This is wire of cytochrome B and C1. Here, a part of this wire is present in intermitochondrial membrane space, intermembranous space of mitochondria. This is cytochrome C. Through this electron passes. and this is also part of wire. Then here the part of wire is this Hem A3, AA3 and copper A and copper B. So you can see copper here. Other places you will see iron, and actually cytochrome has also iron. So here iron sulfur protein, here iron in the form of cytochrome. We will talk about that detail of what is cytochrome. We will talk about that, don't worry. So this is iron and this is iron. This is also iron. Here complex 4 copper. So you can see the wire is made up of iron, copper, at the center organic wire. So this is coenzyme Q is organic wire, and cytochrome C also has iron inside. So you can see iron, and at last copper. So copper wire, copper elements will be at the last, and Hem at the last, copper, and in before copper it also has Hem means iron, copper. It has both iron and copper. So how do you remember the sequence? I will tell you one mnemonics of this. So do you know branch chain amino acid? Branch chain amino acid. So you can remember this mnemonics. BCAA. Branch chain amino acids are leucine, isoleucine, and valine. These are very, very important amino acid and essential amino acid. Valine. So you can remember this mnemonics to remember the sequence of this cytochrome. You can see complex 1 and complex 2 does not have any cytochrome. Cytochrome starts in complex 3 only. So you can see complex 3 B and then C, C1C, 2C. So you can add C1C yourself. So B, C, AA, B, C, C, AA. Here you can see A, A3. So B, C, AA. So if you remember, you can remember the sequence of this cytochrome. Hem is present in cytochrome. Sequence of cytochrome you will remember. Sequence of cytochrome that forms wiring C and C. B, C, C, AA. Now you can see this copper A and copper B. Don't forget this copper A and copper B. It is present in cytochrome 4. This copper is present in cytochrome 4. Here I don't have to explain more. This complex 2 has very similar wiring as complex 1, but it does not pump electron. It is succinate dehydrogenase. So we will go into detail of this. You can see complex 1, because it takes electron from this and then transfer to coenzyme Q. This NADH in due process is oxidized, and this coenzyme Q will get electron. It is reduced. That is why this is called oxidoreductase. So these all complexes are grouped into group 1 of enzyme. They are called enzymes. Okay, they are electron transport chain proteins, are basically enzymes. So you can see complex 1 is NADH coenzyme Q oxidoreductase. What is there? FMN and iron sulfur protein. You can see the complex 2. You know complex 2 is succinate dehydrogenase. Other name because it draws electron from succinate and transfer to coenzyme. That is why succinate coenzyme Q oxidoreductase. Now third complex is coenzyme Q cytochrome C. So third complex you have seen complex 3. It receives electron from coenzyme Q and transfer to cytochrome C. That is why it is called coenzyme Q cytochrome C oxidoreductase. So you already know it has got BC and very different type of iron sulfur protein that I have not shown here. Iron sulfur protein that I have not shown here. So complex 3 also have iron sulfur protein. Okay, that is a bit different. So you don't have to remember that difference. Just remember it also has iron sulfur protein. Now these are wiring. Okay, so you have to understand what is the part of wire through that electron flows. So complex 4 you have got A, Hem A and A3 and copper center. These are the part of wire. So you already know Hem A and complex 4 does not have this iron sulfur complex. Complex 4 does not have iron sulfur complex. Only complex 1 and 2 and 3 has iron sulfur complex. Complex 4 does not have. Now after this, so oxygen will receive the electron, and because of creation of proton, the proton will synthesize ATP through this complex. This is called complex 5. So I have not shown that complex 5 in the picture, though I will talk about later. So complex 5 is called, it is a turbine that we are talking about. It is turbine through which ATP is synthesized? Turbine. So it is called ATP synthesis. It rotates. When proton forms will pass through it, it will rotate. Like turbine rotates when water pass through it. So you can see the structure. You can see the vitamins that are present, vitamins and minerals that are present in that form. Just see the electron transport chain in terms of your nutrition you take. So you can see vitamin B3 is NADH, flavoprotein B2, and iron is iron sulfur protein. So iron, you require iron for electron transport. Coenzyme Q is also a part of. I will talk after this slide. I will talk about coenzyme Q. So cytochromes, it is heme. Okay, heme is also iron. Now copper A and copper B actually it resembles iron sulfur type of protein. So in place of iron sulfur, complex 4 has got this copper A and copper B. You have to understand. So you see copper are there, you see iron is there, vitamin B, vitamin B3 is there, coenzyme Q was there. So these are, so sometimes if there is electron transport chain defect, so you can understand, so which element will help electron transport chain and which element will not help. For example, let's say calcium. Calcium will not help this electron transport chain directly. If there is disorder of electron transport, it will not help electron transport chain. Zinc will not help. So you have to understand, these are the elements that are formed. They form the wire of this electron transport chain. Now, so you can see electron, I have already told you cytochrome C. Just remember for the timing, it is very important for apoptosis. Now you can see these are iron sulfur. So cysteine is an amino acid. Cysteine has sulfur group. So it will, the protein has cysteine, and this cysteine will form bond with iron, and this is how iron sulfur complex will look like. This is for actually box like, and this is 2-dimensional iron sulfur. Different type of iron sulfur complexes are formed in the protein, in the electron transport chain. So in electron transport chain, it has got this protein, and this iron is linked with the amino acids of this electron transport chain. So that, as you know, in our day-to-day life, this covers, non-metal will cover this metal. The same way here with non-metal, non-metal, the cysteine are bound, and it will hold this iron here. This is iron sulfur complex. Okay, just to give, I have given this picture so that you will remember for your memory. Okay, because memory is very important. If you don't remember, you don't do anything. Now this is coenzyme Q. You can see coenzyme Q has this 2 COO group at the end of this tetragonal ring. So you don't have to remember this anything, and you have to remember this. You can see here, actually molecular structure never comes in exam. It is for understand. If you understand molecular structure, you will not forget. That is why I am providing this molecular structure. So this is isoprene, this is lipid soluble molecule, and that is why this coenzyme Q is soluble in lipid. They are present in inner mitochondrial membrane. Now these two double bond O will actually accept electron. You can see first electron will be accepted by 1. This becomes semi, and this becomes quinone. And now again second electron will be accepted by second bond. First by this, and then second by this, and then it will become a full quinone. So this is how it will accept 2 electrons. This is a basically organic part of wiring. So this organic compound can easily accept electron and transfer electron. So these are heme part of wiring. So you can see here at the center of this heme, we will talk about heme synthesis in detail. So at the center of this organic molecule, there is heme, iron, and this iron cannot stay like that. It has to be bound. So it is bound to this tetrapyrrole ring. This ring, any ring with this 4 compound, it's called pyrrole. Okay, this is a 5-membered ring. In 5-membered ring, 4 are carbon and 1 is nitrogen, and this is called pyrrole. If you can see here, everywhere you will see 4 such type of pyrrole: 1, 2, 3, and 4. So this is called tetrapyrrole structure. This tetrapyrrole is with heme, with iron at the center, is called heme. Heme is tetrapyrrole structure with iron at the center. And this heme containing molecules, these cytochromes are heme containing, heme plus protein becomes cytochrome. Cytochrome means heme plus protein. Heme plus protein is cytochrome. So you can say this is protein, this is heme. So this is cytochrome. Why do we name heme C? We sometimes name heme A1, heme A3. So B type cytochrome has different type of heme. So difference will be there in organic part of this tetrapyrrole. You can see side chain here, different side chain is present in heme A, different type of side chain is present in heme B, and then there will be different side chain in heme C. You don't have to, fortunately, you don't have to memorize, just understand this site, why A is given name A3 is given, why cytochrome C is given, cytochrome C derives, has different type of this heme with different side chains. And then interior structure, if you see, all have same interior structure, but side chain varies. So this is why it is called heme. Different, after heme, different letter is added to differentiate this heme from this side chain. So this is heme. These are heme. You can see these are many heme. You can hold cytochrome C as heme. So heme B, C. So you can see here different heme are present in heme A1, heme A3. So these are the wiring of electron transport chain. There comes a cycle called Q cycle. We have seen, I told you in the beginning, a proton is pumped from complex 1 for proton. So actually from complex 1, this protein will pump the proton. But for this complex 3, this coenzyme Q also helps pumping the proton. How? And this is held by Q cycle. So Q cycle helps proton pumping for complex 3. Here also complex 3. The Q cycle helps proton pumping. That is why Q cycle, that is the significance of Q cycle in wiring. So what happens, I will tell you what happens. Let me draw the complex 1. This is complex 1 and this is complex 3. Okay. So in between complex 1 and complex 3, you have coenzyme Q. From complex 1 and ADH, you know, 2 electrons are transferred. First NADH, 2 electrons. Okay, these 2 electrons transported through this, and it will reach Q. And this coenzyme Q will take 2 electrons, 2 negative charge. But after getting 2 negative charge, do you see here hydrogen is being transferred? No, hydrogen is not being transferred. Here is membrane. You see membrane, you will see membrane. This is actually part of membrane. Whole is part of inner membrane. Now what will happen? This coenzyme Q will actually suck hydrogen from here. How many hydrogen? 2 hydrogen. It will suck 2 hydrogen from this matrix of mitochondria. This is matrix of mitochondria. Remember, this is matrix of mitochondria. And then from this matrix, it will suck 2 proton, and then 2 proton will bind this, and this will become QH2. And when it reach here, the coenzyme Q will actually transfer electron only to cytochrome B. Okay, it will transfer electron to cytochrome B, 2 electron only. So what happens with this hydrogen? So this hydrogen will be pumped here. Okay, this hydrogen will be pumped here. And then what will happen next? This here, there is one peculiar mechanism. Only 1 electron is out of this 2 electron, only 1 electron will be transferred to this cytochrome C that is present in inner mitochondrial membrane, inner intermembranous space of mitochondria, cytochrome C. And from cytochrome C, complex 4 will receive this. So only 1 electron. What happens with other electron? Other electron will be given to this cytochrome C that is present, C1, that is present inside complex 3. And this cytochrome C1 will actually transfer it back to coenzyme Q. Okay, this coenzyme Q, and then if you see the same process, if happens from the same process, if you see the same process happening, if this second NADH will transfer again 2 electrons from this, so what will happen? The same, the coenzyme Q will receive 2 electrons, it will be passed, and this will get second, it will get next first electron, it will get from first site, first pathway, first NADH, and second electron it will get from second NADH. So it will now it will also have 2 electrons, so it will draw proton, and again it will reach here. So does it, do you think it making cycle? Okay, do you think it making cycle?
Yes, yes, sir.
Yeah, this is called Q cycle. Now in second step, again you will see when it second NADH transfer this electron, then again there will be pumping of 2 proton. So this is called Q cycle. The significance of Q cycle is proton pump. So proton pump. This question, the question will not come from this. You may not find questions. Sometimes question comes very less frequently from this concept, but you may come across this Q cycle. Q cycle is very famous, so you may come across this cycle. That is why I explained. And now you can see from this. So from as a total, from 4 electron, from 2 NADH, you can see 4 electrons comes here. Out of 4 electrons, 2 electrons will be transferred here, and then 2 electrons transferred to this. Sorry, I told you B, it will transfer to B. Sorry, it will transfer to C, and this will transfer to B. Okay, so you don't have to remember that also, no problem. Even if you forget, anyway, you can understand 2 electron is half is transferred here, half is transferred back to coenzyme Q, and this coenzyme Q again will get, and this coenzyme Q will go here. So this creates a cycle, this is called Q cycle. Now, so we have done Q cycle.
C.
Yes, it is complex 2, and it is complex 2 is enzyme of Krebs cycle, succinate dehydrogenase. Now, so see this. Diagnosed with mitochondrial disorder, which of the following supplement will not be helpful?
A.
Yes.
B1.
A. So you have understood the components of that wiring in terms of your nutrition and in terms of your that structure as well, anatomy as well. So this is the important thing. If you understand the components of this wiring, you will understand the component of this wiring and then flow of electron through the wiring. What is the significance of that electron flow? If you have understood, then it's good. So do you have any question till now?